Welcome back, everybody. I'm Dr. Bobby on behalf of New Age Cosmetic White EdTech Healthcare Consultancy. I welcome all of you back to the regular lectures on biology and chemistry for the exam-going candidates who wish to appear for the NEET exams, All India Institute of Medical Sciences, and for those sitting for the CSIR Council for Scientific and Industrial Research National Eligibility Tests. Today we're dealing with the subject of biochemistry. We'll go into the subject of RNA splicing in particular. So let's start off with a video that explains RNA splicing, and then we'll go into the subject matter proper, right? Okay, here we go. Please hold on.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear viboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Now going into the subject matter proper, variation in the longevity of the mRNA provides yet another opportunity for control of gene expression. Prokaryotic mRNA is very short-lived, but eukaryotic transcripts can last hours or sometimes even weeks. For example, mRNA for hemoglobin in the red blood cells of birds. The process of translation offers additional opportunities for regulation by many proteins. For example, the translation of hemoglobin mRNA is inhibited unless iron-containing heme is present in the cell. There are also opportunities for post-translational controls of gene expression in eukaryotes. Some translated polypeptides proteins are cut by enzymes into smaller, active final products, as illustrated in the figure below, which depicts post-translational processing of the hormone insulin. Insulin is initially translated as a large, inactive precursor. A signal sequence is removed from the head of the precursor, and the large central portion, the AC chain, is cut away, leaving 2 smaller peptide chains, which are then linked to each other by disulfide bridges. The smaller final form is the active form of insulin. Gene expression can also be modified by breakdown of the proteins that are produced. For example, some of the enzymes involved in all cell metabolism are broken down shortly after they are produced. This provides a mechanism for rapidly responding to changing metabolic demands. Gene expression can also be influenced by signals from other cells. There are many cells in which a signal molecule, example, a hormone, from one cell binds to a receptor protein on a target cell and initiates a sequence of biochemical changes, a signal transduction pathway that results in changes within the target cell. These changes can include increased or decreased transcription, as illustrated in the figure below. There's a signal cell, and that's a target cell with a signal molecule going in for the receptor protein, which relays the proteins and activated the transcription factor, where the translation goes in for a new protein. Going into the subject matter again, the RNA interference system, RNAi, is yet another mechanism by which cells control gene expression by shutting off translation or mRNA. RNAi can also be used to shut down translation of viral proteins when a cell is infected by a virus. the RNAi system also has the potential to be exploited therapeutically. RNAi. Some RNA viruses will invade cells and introduce a double strand, which will use the cell's machinery to make new copies of viral RNA. And viral proteins. The cell's RNA interference system can prevent the viral RNA from replicating. First, an enzyme nicknamed dicer chops any double-stranded RNA it finds into pieces that are about 22 nucleotides long. Next protein complex is called risk RNA-induced silencing complex. Bind to the fragments of double-stranded RNA, winds it, and then releases one of the strands while retaining the other. The risk RNA complex will then bind to any other viral RNA with nucleotide sequences, matching those on the RNA attached to the complex. This binding blocks translation of viral proteins at least partially, if not completely. The RNAi system could potentially be used to develop treatments for defective genes that cause disease. The treatment would involve making a double-stranded RNA from the diseased gene and introducing it into cells to silence the expression of that gene. And the RNA interference system is also explained more completely in the video below from Nature video. Let's check this video as well, right?
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand.
The combination of the RNA and Argonaut, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaut catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonauts and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
ഇത് ഗ്രഹപ്പഴാണെന്നുണ്ടാ ഫുൾ ഗ്രഹപ്പഴാണ് ദിവസം. പക്ഷെ എന്തൊരു കഷ്ടമാണ്.